we've been using in vivo delivery of EVs during the last years, and we identify those organs who EVs are reaching right after the tumor is circulating. So really, since we work in this pre-metastatic niche setting, we label them with fluorescent dye, we inject them in mice in preclinical model, and we see the biodistribution in the different organs. We saw that depending on the tumor organotropism of metastasis, so imagine you have a lung-tropic tumor, so they normally metastasize in the lung. We saw that these EVs carry a specific determinants, such as integrins, for example, that they actually make them to home specifically in those organs. And in the last work I mentioned in the lymph node, we saw that there are specific markers in the lymph node, well, in the EVs, who basically lead them to the lymph node and modify the behavior of resident cells. So we see how they specifically reach those organs and which are the main cell type affected through the EVs. We saw the concept, we call it education, because we saw that determinants of tumor-derived EVs are changing the behavior of resident cells. In the lymph node, for example, lymphatic endothelial cells, we saw that are the main ones. And in the lungs, we saw that there are other populations affected, not only locally, but also distally, such as bone marrow-derived cells. That was my main work in EVs, how these EVs can reshape or remodel the microenvironment. And the outcome of this is they normally collaborate with metastatic outcomes. So the tumor-derived EVs reprogram this microenvironment, and these factors promote the metastasis later. So it's a pro-metastatic effect. And this is how we are starting. Luckily, we were able to define not only those markers, but also novel therapies, such as, we can block integrins, or in this lab later work that we are going to publish, we found a specific protein called NGFR, the receptor of NGF, that could be specifically, targeted with local therapies. And we hopefully will find that this is in combination with other active drugs used in the clinic, a good combination to prevent metastasis. And hopefully we saw also like immune cell reactivation, no?